So looking forward, things that are due soon is the next muddiest point is, of course, due this upcoming Sunday. The mini project for Unit 1 on the genetic algorithm is due this Sunday, and that's one that is not dropped. And that's the only one that is not dropped. And so I want everybody to at least have that experience. And it's a relatively, I think, low key assignment asking you to build a very simple genetic algorithm, make a couple very minor modifications to it, just to experiment with some of the key operators in the genetic algorithm. We're moving on into Unit 2, and so there will be a concept check on Unit 2, not due this Sunday, but next Sunday. So Unit 2 will be today, Tuesday, Thursday of this week, and then Tuesday of next week, and then we should be done with Unit 2, so then we can do the concept check on Unit 2 for this next Sunday. All right, so if you have any questions or issues with the deadlines going forward, just please feel free to send me a note. Moving forward then, like I say, we're starting into Unit 2. We're sort of going to pivot from where we left off in Unit 1. So in Unit 1, we were talking about genetic algorithms, and we didn't present them quite this way when we stopped with Unit 1. But if you really think about it, genetic algorithms are sequential sampling methods that adapt their bias. And so what we mean by that is that we're using the population, the individuals that are generated to sample the fitness space, and then from there, use that as evidence for how to adapt their bias in where they're sampling. So the actual sampling approach, once you sort of are centered, doesn't change. You mutate the same way, regardless of where you are in the genotype space, in your decision space, in your solution space. But you use the progress that you've experienced based on the past solutions to decide where you're going to do that sampling. And then again, once you do that sampling, you start mutating and so on, that mutation rate is sort of fixed. It's not dependent upon the fitness function. So we're adapting our bias for our search through the space, but not our variance. But we can view it broadly as sequential sampling, in that we're sampling from an area using the results from that sampling process to then affect the sampling in the next iteration. And so the hyperparameters of the GA are really changing how the evidence is used, or what the algorithm sees with that evidence. And so in fitness proportionate selection, FPS, the idea here is that we're following fitness changes of at least 1 over R in terms of relative fitness. So there's this band. So if you're sampling a region of the fitness landscape, and you have a bunch of samples that are going all over this fitness landscape, then if you were to look at that population, then if any fitness differences that are inside of a 1 over R band of relative fitness, so if you took the average fitness in that whole population, then any individuals that don't differ by more than 1 over R end up not being seen, or you lack a sensitivity to follow variations that are smaller than that. So individuals that do not differ that much in fitness, the differences between those individuals are not going to drive changes in the bias of the sequential sampling algorithm. So by changing the number of parents, changing the R, then you end up making the algorithm more sensitive to small changes. And so smaller changes in fitness will end up causing the next generation to kind of be locked into those smaller changes. And so that makes it a more higher precision search that will linger around the same areas of the fitness space for longer. So we can kind of say, well, at equilibrium, then what can we infer from the population? Well, the big point that I'm going to get about these GAs here, which will be different than these methods looking forward, is that we can't really tell a whole lot about the population. The population isn't that informative, but if we were to try to squeeze as much information out from the whole population rather than just the best individual in that population is that genotypes that differ are only going to have meaningful differences if they differ by outside of this 1 over R band in relative fitness. And so the idea here is if you have very few parents in the population, then if you look across the individuals that come out at equilibrium at the end of the genetic algorithm, then you might get a bunch of fitness variations. And there might be a top individual. Of course, there's going to be a top individual. There has to be. But if you were to look across the genotypes and say, how do changes in the genotype predict changes in performance, then individuals that are very close to that top individual, changes in their genotype relative to that top individual are basically noise. But if you look outside of a sufficiently large band to individuals that are much farther from that top individual, then the differences in their genotype relative to the top individual are much more predictive of causative real likely causative changes in function. So the more parents you have, then that basically makes more of those individuals meaningful because that band of indiscriminability becomes smaller and smaller. But really, what you're doing when you're changing hyperparameters like R is you're telling the search how to focus its search from one generation to another. You're not really doing something that gives you a lot more information at the end of the genetic algorithm's many generations when you look at that whole population. So that whole population is kind of interesting because it gives you a top individual and a bunch of like alternative candidates in case your stakeholder really doesn't like the top individual that the algorithm has picked. But it doesn't quite tell you about how the structure of the population, how that lets you infer a whole lot about the genotype changes themselves. If we switch to a different selection paradigm, like rank-based selection algorithms, it does change the focus of the algorithm as it moves from generation to generation, the sequential sampling process. But again, it's not going to really make the population at equilibrium much more informative. It's you can try to take something from that final population, but there's not a lot there. So if you flip to the rank-based selection methods, you're going to regulate selection pressure. So what we mean by that is if you have large fitness differences, the rank-based approaches equalize all those. They bring those together so that if some individual or candidate solution has a really high fitness relative to everybody else's low fitness, then that gap between those fitnesses doesn't matter anymore. Only the ordering matters. So it becomes an ordinal response variable. You're interested more in the order statistics than the absolute fitness statistics. And that sort of changes the sensitivity of the algorithm. Now, what's interesting is it makes the algorithm more sensitive as fitnesses become much more similar. So as fitnesses is coming together where you really can't tell that much apart. So where fitness proportionate selection might start viewing everyone as having equal fitness. Well, then rank-based selection, it doesn't see those fitnesses coming back together. It still sees the ranks. And it will still focus on that top ranked individual. And then based on what you've chosen R to be, then it will extend its focus. It will keep other individuals in and more. So the larger you make the R, the more it keeps lower ranked individuals in the population. But still, it doesn't really help us in making sense of the equilibrium population at the end of the algorithm. It still ends up kind of being, well, we focus primarily on the top ranked individual in the population. And then if someone asks about the lower ranked individuals, then what we can tell them is based on our selection of the R hyperparameter, there's only kind of meaningful order differences in the kind of top ranks. And so the smaller the R, the less you can trust any of the orderings kind of at the bottom of the list. And so if you notice regularities in the genotype, regularities that you see at the top of the ordering maybe are meaningful, but at the bottom of the ordering are pretty much noise. So that's kind of how R affects that. But in both cases, really, that hyperparameter is affecting the focus of the search. So how the sequential sampling method goes from one generation to the next. You're just kind of giving it a different lens to look through as it samples the space and then uses that as evidence to adapt its bias. But again, I want to drive home here, we're not adapting the variance. The way it samples stays the same once it's kind of centered, but that choosing where that center is going to be, that's sort of that's the bias, and that is being affected by the populations. So that's kind of like our GA. The population guides where to center the next step in the search, but not really how to sample at that location. And so that opens up a motivation here for, could we adapt the shape of the population so that in certain regions of the space, we're more likely to mutate in some directions and less likely than others? And that really, that idea is something that motivates this other strategy that we're going to be talking about today, evolution strategies, or ES. And so with the GA, the population is a mechanism, not an output. The population is just a control surface. The population is how the samples are driving changes in the bias. What we would like to move to in evolution strategies is a way for the population itself becomes almost an output. It carries the shape of the population, carries information about the underlying fitness function, and we can use that to make much more efficient searches where we don't just search everywhere in every direction, we search in the most informative directions. All right, so that's the motivation for today. So going back in history, genetic algorithms, the kind of most pure earliest form of the GA was developed by John Holland. It was a lot simpler than the GAs that we've talked about. It had sort of fewer components, fewer degrees of freedom, and those limitations allowed for Holland to build this so-called schema theorem that allowed him to make proofs about the way the GA works. Under what conditions could we guarantee that the GA reached an optimal solution, and so on and so forth. In biology, this has connections to the so-called price equation, which tells you how traits evolve from generation to generation. And that's great. That analysis, that pure version of the GA and the analysis came out of it was really beautiful, and it gave a lot of interesting insights into how GAs work from a kind of a theoretical point of view. So they had a lot of analytical tractability for studying them. But as people played with the GA, we got the more modern versions, like the ones that we've now presented in the past unit, and they have much more powerful hyperparameters, but they basically wrecked the analytical tractability. So with a modern GA, we know empirically they work very well, especially for certain types of problems, but we can't guarantee that they're always going to find a global solution or anything like that. So we don't really have the same mathematical guarantees, but we get much better performance. We know that the GAs work for a wide range of problems. They work for problems with integer decision spaces. They work for problems with discrete decision spaces where your decision variables don't have a natural order to them. They work for real and they work for mixed versions of these things as well. So this is a very general purpose approach. And so that's what we got out of the GA, is a nice global optimizer that works for a general set of complex multifaceted objectives. Started out with theoretical guarantees, and then now we have modern versions which have more empirically demonstrated performance on this wide range of problems. So let's pivot then to around the same time that Holland was working on the GA, there were folks overseas, say in Europe, that were working on evolution strategies, which is a fundamentally different way to think about searching through these spaces. And evolution strategies, like I said, it's a totally different evolutionary algorithm. We're going to give up on some of the generality that we get in the GA. So we're going to focus on totally real valued chromosomes. So there are ways that you can think about adapting modern evolutionary strategies to use integer valued genotypes, but they're heuristic. And so they don't quite have the same interpretation. So we really sort of, in order to get the most bang out of our buck for the evolution strategies, we have to stick with the real valued chromosomes. There is crossover. There are both types of, there are crossover similar to what we see in the GA, where you get discrete swaps of genes, but crossover is most commonly through linear combination, which goes along with these real valued chromosomes. The big change with the evolution strategies is that now we actually mutate the mutation rates themselves. So we view the mutation rates as step sizes, which are part of the chromosome. So the chromosome includes your decision vectors, and it includes step sizes for each variable in the decision vector. And then those mutation rates actually get updated themselves through a hyperparameter that you set. So you've got one or two hyperparameters which mutate the mutation rates, and then the mutation rates are applied after they're mutated to the decision variables. So every individual has a different mutation rate. And this allows for you not only to be searching through the solution space, the candidate solution space, but it also allows you to search over ways to search in the solution space. That's kind of the key here, is that it allows for variance adaptation. So you can say, in this space, we notice that mutations of these decision variables that are larger tend to give us more increases in fitness than the same size steps in another decision variable. So that might mean that we're going to mutate one decision variable more to walk more quickly through that dimension, and another decision variable less. So we're going to be more sensitive on mutations for some decision variables than others. And theoretically, you can even imagine linear combinations of decision variables. Those could be your mutation directions. So we're going to get into all that and see what that looks like here. But that's kind of the key idea, is that we're going to adapt our step sizes. We're going to adapt our variance based on our progress so far. So we're rewarding not only the decision variables, but we're rewarding the mutation rates too. We use deterministic selection in these approaches, which effectively eliminates drift. So the key point here is that because we're adapting the mutation rates based on our prior successes, the resolution of the algorithm is going to be set up through the mutation rates and how small those step sizes are and how big those step sizes are, which are self-tuned, and not through the number of parents. So in the GA, the number of parents kind of set the resolution of the algorithm. But in the evolution strategies, we can get away with having just one parent, because that one parent is going to have mutation rates which can be adjusted. And so that one parent can make small steps from parent to offspring, or very large steps, or a mixture of small steps in some directions and large steps in other directions. So that manages to store discovered info about our optimization objective into the genotype that's not stuck in the population demographics. And so that's why in ES, it's not uncommon to only have a single parent. So overall, to take all of that together, and the goals for the evolution strategies are fine-tuned searches that are guided by discovered fitness shape. So you could say that the intermediate goal is to not only discover high fitness regions, but discover how those high fitness regions change over changes in the genotype. So it's discovering the kind of partial derivatives or the geometry of the fitness landscape. And then using that geometry, it then creates a highly efficient search where you can concentrate mutations in the most informative directions. And that should hopefully allow for a fine-tuned exploration of a local space that uses fewer samples to get there. So the GA goal is very much focused on global. The ES goal is going to be focused more on local, at least initially. Okay, so let's see what that looks like. So a basic, pure classical evolution strategy, ES, we use certain notation to give us most of the hyperparameters in a compact way. Now, remember, most of the evolution strategy is doing a lot of self-tuning behind the scenes. So we're going to be talking about far fewer hyperparameters for an ES than we would for a GA. So, and it's more about the structure of how those hyperparameters interact and less about how many hyperparameters we have. So these are the two basic forms that we start out with the ES. And so this mu, you can think of a little bit like in our GA notation. This is a little bit like R comma M, where R is the number of parents and M is like the number of the population size. And so and it just happens to be convention that we use mu as a representative of sort of the number of parents and lambda as representative of the number of offspring. So the way we read this mu comma lambda is that this ES is configured to have mu parents, each one of those parents then produce, not, I would say, and then there are lambda offspring produced by these mu parents. So you can think that let's say there's going to be lambda over mu offspring on average generated by each parent. So you can think that over the set of these many parents are going to contribute equally to produce these offspring. So if you need to produce, let's say this is 3 comma 9, if you need to produce 9 offspring, then you're going to iterate and say, okay, we're on offspring 1. Well, from offspring 1, we're going to choose randomly among these 3 parents. So remember, this is 3 comma 9. So the first offspring of these 9, we're going to choose randomly from these 3 parents to be the source of that. And then we are going to then take that parent, and remember that parent has mutation rates. Well, we can apply the hyperparameter to mutate the mutation rates for that parent. And then once we have new mutation rates for that parent or for that parent's kind of proto offspring, we can then apply those mutation rates that are in the genotype to the new individual itself. And that will produce a new offspring that will be one of these 9 offspring that will be similar to that randomly selected parent, but then mutated in that way that was encoded in that genotype. And then we do that over and over and over again for all of those 9 offspring. So we have mu parents to produce lambda offspring. And then after we've chosen lambda offspring, then we need to then figure out which parents do we use in the next generation. Well, in this comma here approach, so mu comma lambda, that means that once you've generated your lambda offspring, so let's say your 9 offspring, then you're going to discard the old parents completely. And then you're just going to choose the best mu from that lambda offspring. So if this is 3, you'd choose the best 3 from those 9 offspring. And this is a completely deterministic choice. There's no drift. This is like ranking selection with stochastic universal sampling, kind of. But I mean, it's just totally deterministic. Just rank all of those offspring and take the top 3. And those become your new parents. And then you can do that over and over again. An alternative form is to introduce elitism into the ES. And that is implemented with this plus here. And that means we do everything the same, but we then choose the best mu from the combination of the old parents plus the new offspring. So if this was 3 plus 9, then we're going to build 3 parents. So this is 3, let's see if I can do this. 3 plus 9, then we're going to choose 3 parents and produce 9 offspring. And then we're going to choose the best 3 from the 12 that are produced, where we have the 3 old and the 9 new. And so that's got this elitism. This helps ensure the best fitness stays in the population. So that's a reason that you might do that. So examples, we might see 1 comma 6. you've got one parent produces 6 offspring, and the best one of those offspring survives to be the next parent. 2 plus 3, we've got elitism there. We've got 2 parents. Those 2 parents produce 3 offspring, so each offspring can be viewed as coming from one of those parents at random, and then it gets mutated. And then once you have those 3, you add those 3 to the original 2, and then choose the best 2 to become the parents in the next generation. Likewise, 5 plus 2, you could have 5 parents. So lots of parents produce 2 offspring. But then as long as you have elitism, then you're going to get a population of 7, and you'll be able to draw the best 5 from that 7. You cannot have 5 comma 2, even though you can have 5 plus 2. And you might think about, well, why is that? Well, if you produce 2 offspring, what this is telling you is that those 2 offspring need to then, you have to take the best 5 out of those 2, and that's invalid. So 5 comma 2 is one of these invalid combinations. So here, the number of parents you have here is going to affect the speed of the search. Well, I guess it really, all of so whereas in the GA, we were worried about resolution and speed. So we might want to increase M, but then once we increase M, that changes we have a certain computational budget for M. We can then trade off, we can convert, you can decide between convergence rates and things like that have to do with how we choose R. And then we can also choose mutation rate, and that also affects things. There's a lot less to worry about here. Here, the resolution is set by the step sizes, but the step sizes are automatically adapted within the algorithm. So we don't quite worry about the resolution of the algorithm. So all we're sort of left with is the kind of speed of this algorithm. And so we have a certain number of samples that we're sampling of the space. And then we do have some retention of those samples. And that is going to affect how quickly we move through the space, but it's not going to affect the resolution. So this doesn't act like R in the GA in quite the same way, because there's no 1 over R that you have to worry about. All right. So key point here, that mu sets the evolutionary inertia like R, but it doesn't set the resolution. And mu always has to be less than or equal to the lambda or the mu plus lambda. So if you're doing a comma, then mu has got to be less than that lambda, because you need to select the next mu from the lambda. All right, so that's the simplest one. Now we can add in recombination. And so with recombination, then we're adding this here. So this will be like this will be like 3 parents, and we're going to recombine 2 of them and produce 9 offspring from that. So that would be one way, or it could be 3 comma 2 plus 9. So the plus and the comma work the same way as they do up here. And the way recombination works is that you select row parents from the mu total parents at random. And then from there, the offspring can either be, if you're using intermediate recombination, and usually an author who is publishing results in this will say what type of recombination they're using, or they'll indicate it with an I, a capital I subscript on the row. So it might be like 3 capital I, like that. And so in intermediate recombination, you're taking the average. So you're just taking an equal weighted average of the row parents. And so that's what you would start out. So your offspring is going to then, you're going to say, all right, so if I've got 4 parents and I'm doing a recombination of 2 of them, then I select 2 of those 4 parents at random, and then I take the average of those 2, and that ends up becoming one of my 9 offspring to start with. But then I go through the mutation process after that. So once I've generated that one offspring, then I mutate its mutation rates using that upper level hyperparameter, which I haven't written down here. And then once I have new mutation rates for that offspring, then I use its own custom mutation rates to mutate its decision vector. And then that becomes my new offspring. Now, I could have done recombination in a discrete way as well. So the discrete with the subscript D says that I take those randomly selected parents, so 2 parents or 4 parents or whatever, and then for each allele, or I guess I could say for each gene in the offspring's genotype, then we're going to select an allele at random across the number of parents we have. So in both of these cases, if this was a one, so it would be a little silly to put a one here, but technically it's legal. You get rid of recombination entirely because the average of a single parent is that parent itself. Or if you were choosing an allele at random from a set of one, you're just going to get that one allele. So if you do /1 here, you basically get this case back. But if you do /2, /3, and so on, then you're either averaging that number of parents or at each location along the genome, you're choosing among that many alleles at random. Okay? So examples down here, 5/2, 6, meaning we started out with 5 parents. For each one of these 6 offspring, we chose 2 parents at random. And if they don't give us any other information, we can maybe assume intermediate recombination, which means we're going to take those 2 randomly selected parents and average them together. They're just real valued chromosomes, don't forget. And that will produce an average real valued chromosome, which will be one of these 6 offspring. And then we can then do the mutation process, which again means we're going to take the step sizes that are in the genotype for the offspring, mutate them with a single hyperparameter, a single, or sometimes there's 2. It's kind of they're basically learning rates, and so you can have kind of a first order and a second order learning rate. Those can apply to all of the step sizes for that particular offspring at once. And then once you have new step sizes, which remember 1 step size per decision variable, then you can apply those step sizes for that single offspring to its decision variables. And then so at that point, you'll have new decision variables with new step sizes, and that will be one of those 6 offspring. And then you repeat that for each one of those 6 until you get all 6 offspring. Once you have those 6 offspring, then you draw the top 5 in terms of fitness. And those then become the parent pool for the next iteration of this, and you repeat. 4/3 + 1 is similar to that, but we're going to say we have 4 parents, or we produce 1 single offspring. That 1 single offspring is going to start out as a recombination of a random set of 3 of these 4 parents. And once we have that 3, then we can average them together, and this is just the one average. Or we can do, if the author said they use discrete recombination, then swap, choose a random allele from each one of those 3 parents. And that's how you start out with that offspring, and then you go through that mutation process. Once you're done generating that new individual, then you add that back to the pool of the original 4 parents plus that new individual. And then of those, you see who are the top 4. And those top 4 become the parents in the next generation. The other things you can do is to be more explicit about what type of recombination. So this is saying I'm going to choose 4 parents for recombination, but we're going to do discrete recombination, or I'm going to select 2 parents and I'm going to use intermediate or linear recombination. So that's how we do the basic notation, and we add on recombination. Now, note that the chromosome is split into the decision vector and the step sizes. So you can think of decision vector as X and step sizes sigma, which you can think of like as a standard deviation. Okay, and so so keep that in mind. Now, oh yeah, and I just want to say, as an interpretation, almost forgot about this. As your recombination hyperparameter gets up to your number of parents, what recombination is doing is it is contracting your population more and more. So remember that like in the intermediate, we are literally taking an average. And so if you set your recombination parameter up to the number of parents, then all of those parents, they're scattered around the decision space. Then all of the offspring are just going to be an average of all of those parents. So they all are going to start before mutation in the center of mass of all of those parents and then mutate out from there. So that will contract the population so that the offspring are all in the middle, and then mutation might shift them out again. Discrete shuffles. So, but which is kind of a similar idea. So if you took rho and made it as big as mu, then what you're basically saying is that all of your offspring are just going to be a random reshuffling of all of the alleles in all of your parents. So it's sort of a discrete version of contraction where it's very explicitly contraction in the intermediate case. All right. So that's recombination, but I want to focus on this over here for a second. So again, our genotype or chromosome is split up between the decision vector and the step sizes. Both of those right along with every individual. So let's step over here. And if we think about that, chromosome is split between those. We can apply different recombinations to each. So for example, you could apply discrete recombination for your X values, but intermediate for your mutation rates. Because it doesn't really, I mean, it's rare to, it's hard for me to come up with a good example of why we might want to use discrete recombination for the step sizes. The step sizes, it's I think I mean maybe there are good examples for that, but step sizes approximate a gradient, and so maybe a smooth average update makes more sense than just swapping a step size with another individual. So you might decide that you want to use the averaging recombination, the intermediate recombination for the step sizes, but the discrete recombination for the decision variable, a decision vector. So you can do that by splitting up your recombination, and usually it's in this order, where the decision vector comes first and then the mutation rate part comes second. So if you use this here, then this is telling me I have 6 parents, and those 6 parents, I am going to use a recombination of 2 parents with discrete recombination for the decision variable part. But for the step sizes, I actually want the step sizes to come from all of the parents, and I want that to be intermediate. So this is my decision vector is going to have more diversity. It'll have less contraction than the mutation rates, but the mutation rates will gradually be averaging from all of the mutation rates. So you can imagine that this will maybe create a slowdown or a pressure for slowing down changes in the mutation rates, while it still allows for more variation to exist in the decision vector itself. So that's one example, and then you could do that with elitism. So here's a case where I'm using 2 parents as the source of both of those for the decision vector and the mutation rate. So the point is, you can separate out step sizes and the decision variables, and you can say, I'm going to use recombination one way for one and the other for another. And it's not just in the type, discrete and intermediate, but even in the number of parents. All right. So just keep in mind that as we're looking through these ES, overall, ES is doing 2 things at once. Because it's doing so much with these step sizes, over time, those step sizes are going to capture the curvature around the individual. So you basically can imagine that if you have individuals, like 2 individuals that are at the same point in the decision space, if one individual has very large step sizes that make it so that that individual's offspring go to a vastly different part of the fitness space, where it's far less fit. But the other individual has step sizes that cause it to move only in directions of increasing fitness. then the one that is moving in directions of increasing fitness will tend to be more at the top of the list. So we're not only capturing the instantaneous position, but we're sort of capturing the velocity of these individuals that are moving through that. And in doing so, the step sizes are capturing curvature around the individual in the fitness landscape. So the focus here is on the kind of exploitation and fine tuning around those individuals so that we can exploit directions so that we can tune the exploitation so that we can take very, very tiny steps when it's important to exploit, and very kind of larger steps when we have no evidence that there's small scale variations for us to exploit. So it allows you to start kind of to explore in some dimensions and exploit in others, or just generally balances the exploitation, where exploitation may require large step sizes in some directions and small step sizes in others. So that's the first thing it's doing. And then once it kind of understands the curvature there, then it samples from that curvature to generate high fitness examples. And so this information accumulates over time in these step sizes, not in the population. So mu equals 1 can be effective, as I hinted at before. So again, we don't have to worry about things drifting off and us having small populations that accidentally get lost to drift. So what we're seeing here is that we're using information from mu individuals to update our model of the curvature of the fitness landscape. And then once we've built a new model of the for curvature of the fitness landscape, we then take lambda samples from that model, and that just tries things out. So we're probing with these lambdas. So this is sort of a probe vector here. So how much information do we want in the probe? How much computational budget can we put into probing this newly modeled, this hypothetical fitness landscape? And then once we've gotten all of that data, then we can take the top individuals from that and then use that to update our model. So we update with this portion. So that's really the reason why ES is split into these two parts. It's how much effort do you put into probing and how much effort do you put into model updating? That's what's going on here. Those are the two parts that are happening here. All right, so that's the traditional evolution strategy. These ideas, this is what motivated the production of this ES at first. Is how do we make more efficient, fine-tuned search? So it's a very different focus than the GA. Now, over time, people figured out other ways to do this that started looking a bit more Bayesian. It almost looked like they had priors and posteriors that were being updated. So here, the model is implicit. It emerges from the step sizes. So the step sizes somehow encode information about the fitness landscape, but we aren't actually using a low order model in order to really capture a model of that local fitness landscape, not in at least pure evolution strategies. So that brings us to natural evolution strategies, or NES. This is toward the so-called CMA-ES, covariance matrix adaptation ES, which will be the next thing we talk about here. But on the way to that, this is kind of like, whereas CMA-ES, which is like very successful and used, it's a modern algorithm, it doesn't have as many theoretical guarantees as NES. So NES is kind of like that pure GA that Holland developed, and CMA-ES is kind of like the ugly GAs that have been developed since then, that despite being ugly, perform really well. Okay, so the NES is a special elaboration of evolutionary strategies. It constructs a distribution whose parameters are updated based on the fitness of samples from that distribution. So we are explicitly, when you do an NES, you set up that distribution ahead of time. You say, I'm going to use a multivariate normal, a multivariate Gaussian. That will be what I'm using to model these local shape of the fitness landscape. And then I'm going to update the mean and covariance matrix of that over time. But you can use almost any distribution with NES. And NES just provides a way to update that. Now, those of you who are familiar with good old-fashioned AI and traditional machine learning, if you've taken classes on good old-fashioned reinforcement learning, you may have seen some of these concepts before. You may be familiar with the natural gradient, which we're going to see in a second, is where this natural comes from. And so all of those ideas that went into some of those traditional RL and some other aspects of machine learning too are going to were highly motivational in setting up this architecture too. So let's see that. So the idea here is the distribution you choose, which is a little bit like a prior, like a Bayesian prior. It is reshaped to match the apparent underlying structure of the optimization objective as you sample from that prior distribution. So you sample it, you see what information you got back from that. From that, then you try to update that prior to be reflective of the information that you received from those samples. So you're kind of updating these expectations to kind of minimize surprise over time. That's kind of the idea here. So I mentioned surprise. Whenever we hear the word surprise, when we're talking about machine learning, we should think about information, because information is how we quantify surprise. So this comes from information theory. When we get to thermodynamics in a couple of units, we'll make this a little bit more explicit for those of you who aren't as familiar with information theory or with the connections with surprise. But for now, I'm just going to sort of throw out this term natural gradient, which comes from the field of information geometry. And this is a quantity capturing the shape of statistical models. So what we mean by that is there's this tool called Fisher information, which quantifies which directions through search space provide the most information. In other words, the biggest changes in distribution. So it's this idea that if you've got a bunch of decision variables, which are just directions in your decision space, then if you imagine you're at a flat spot in the fitness landscape. So you've got this fitness landscape that's relatively flat down here. Well, if you were then to move in this direction, then you're not gaining a lot of information taking small steps in this direction because it's so flat. You have to you sample a little bit farther away, and you're going to get effectively the same fitness. And so you have to sample much farther away in order to start seeing changes in the distribution. Now, that might be in one direction, but in another direction, so coming out of the page, it might be much steeper. And so in that case, then if you take a small step, you do get a large change in fitness. So coming out of the page, you get large changes in fitness going with the page. So going with the page, you get small changes in fitness for the same step size. Coming out of the page, you might get large changes in fitness for that step size. So Fisher information helps tell you about what directions give you the biggest changes in information. So it's the most sort of, if you want the most surprise, if you want the biggest change in fitness, then that's going to come going in this particular direction. So you can define this so-called natural gradient, which will give you that direction through the decision space. And it's actually through the step size space too, that tells you if you were to change these variables in this particular direction, that's going to be the most productive way to increase your fitness. Now, that doesn't tell you how big the step sizes are going to be, but it tells you where to place those step sizes. So once you know the gradient, the uphill, then from there, you can scale the step sizes to be smaller for the informative directions and larger for the less informative directions. So you can rapidly go through the flat parts of the fitness landscape. You run through that as fast as possible. The steep parts of the fitness landscape, you kind of move more slowly. Because if we're talking about like fitness peaks rather than troughs, if you're moving in a direction like this, you want to take small steps so you don't overshoot this peak up here. So if you're getting major increases in information or in changes in the fitness distribution, as you move in this direction, then you're going to take small step sizes. And if you instead have a fitness peak that looks like this, then if you take small step sizes, it's not very informative. The fitness isn't changing that much. So you're going to take large step sizes in that direction. So you assign the step sizes, you kind of smear them out along the natural gradient, which comes from this Fisher information matrix. And that, like I said, that natural gradient points towards your locally higher fitness, with step sizes compensating for this information curvature. And, so where does the natural come from? Notice here that our optimization objective, I haven't said that it has a gradient, and it doesn't. So this natural gradient it doesn't have to. This natural gradient does not require a differentiable fitness objective. That's not what it's doing here. It also is invariant to parameterization. This is what's so important in other machine learning applications too. So regardless of how you write your fitness function, regardless of how you choose the dimensions of, whether I'm using a parameter in inches or meters, those decisions of how you set up the coordinate space do not change the direction of the natural gradient. So this is a gradient that is purely in terms of information value and information gain. And it will automatically scale and rebalance for whatever coordinate frame you choose to make sure that you're always going in the direction where changes provide you the most increases in information or are most likely to increase that. That's how I should say it that way, are most likely to increase the fitness objective as you're sampling. So you sample, you take a bunch of points, looking at those points, then this Fisher information stuff and this natural gradient is going to say, based on these points that you found, then the most likely way to increase your fitness is to move in this direction. And that's going to mean change this decision vector a little bit, this decision vector a lot, and so on and so forth. This step size a little bit, this step size a lot. So it can affect the mean and the variance of this distribution. So that's kind of a key point here, is that remember that this is a parametric algorithm, this NES. So the step sizes are affecting the variance parameters of this distribution. Back in normal ES, the step sizes were literally step sizes. And so they were literally the spread of how you're sampling through the space. Here, the parameters themselves, so the variance parameters of the distribution you chose, like you might have chosen a normal distribution with a certain mean and a certain variance. And so that we can think that we're trying to update the mean and the variance of that distribution in those two things are being stepped through the options that you can do for these things, so that the next samples from that normal distribution are going to then give you information about how to continue to change those steps or change those two parameters to move towards higher and higher fitness. So the idea here is that we are trying to adapt the parameters of this distribution to fit the optimization objective so that we can predict the best way to move. And because we're just changing this distribution by changing its parameters, then we theoretically can actually change the population size as we go. So I'm just going to be talking about cases where the population size is static, but you could theoretically say that, wow, we're in a really interesting part of the fitness landscape. Maybe locally, we're going to increase our lambda, increase the number of samples that we're going to generate in order to gain information about how to change these parameters. So you can change population size flexibly over time, dynamically, because of how this algorithm works. Okay, I think that might be that was a little hand wavy, but I think that when we talk here about CMA-ES, which is sort of the big important part that we've been building up to, then I think it'll start making some of that NES click. I don't want to drill down into the details about how NES works, and I'm not going to drill down in a great deal into how CMA-ES works, but I at least I'm going to try to make things a little bit more concrete with CMA-ES. So CMA-ES is closely related. It's meant to sort of be an approximation of a natural gradient system here, or of a natural evolution strategy. It does not use the formal natural gradient, but it uses a heuristic approach to, I guess, a meta-heuristic approach to try to emulate what a natural gradient is trying to do, but to do it in a way that is a little bit more fast, a little bit more efficient, and so on. And it turns out that CMA-ES ends up has shown itself to be very, very good at what it does. So most people who use evolution strategies, when you talk about modern evolution strategies, typically you're talking about CMA-ES. All right, so how does CMA-ES do or work? Like I said, it does not use a natural gradient, but it's a similar approach. You can think of CMA-ES is to NES as is the modern GA is to Holland's kind of pure and simple GA with its schema. So you get rid of your principled mathematics, but you gain proven empirical success. So the way CMA-ES works, I'm going to just switch to R and M here. Before, remember, we used mu and lambda in ES. Because the distribution that is used in CMA-ES is itself a multivariate normal or multivariate Gaussian, then the mu becomes a bit overloaded. So I'm going to be using mu in that distribution, and that's why I switched back to just using R and M in this description of CMA-ES. So the idea here is that CMA-ES, every iteration is going to take M samples from a multivariate normal distribution whose parameters are shaped by the R best samples from the previous generation. Specifically, at generation K, we're going to generate a population, so basically samples here, from this multivariate normal, that has a certain mean and a certain covariance matrix. So as a reminder, the covariance matrix is going to have down the diagonals, it's going to have the variance in every dimension of this normal. And in the off diagonal, it's going to capture correlations between the dimensions. And so how the dimensions covary. So in a multivariate normal that is round, so if it's a two-dimensional normal that has a scatter shot of points that just land in a circle, then we've got a variance in this dimension and a variance in this dimension. So those would be on the diagonals. But there is no correlation between the two dimensions because all the points land in a circle, or you could argue land in a square. And so because you can't, a point that's over here is just as likely to be up and I guess I'd say a point that is that projected down onto this dimension is just as likely to be up here as it is down here. There is no covariance between the two of them. There's no correlation between these two dimensions. And so you would have a zero in the off diagonals of that two-dimensional case. But if you instead had a cigar-shaped normal distribution, then so you've got points that show up in this cigar, then you still would have, and maybe it's better for me to draw this cigar like this, then you still have a, okay, well, this is the good point here. If I drew the cigar like this, then I'm going to have a very large variance in this direction, a very large or very small variance in this direction, but again, no covariance because the points over here are just as likely to be high as they are to be low. But if this cigar shape was this direction instead, then the maximal covariance or the maximal variance here is actually oblique. It moves in both of these dimensions here. So you can quantify the variance. Like if you take this cigar, you can project it down to this dimension, and you can figure out the variance along this individual dimension. You can also project it this way along this dimension, and you can talk about the variance in that dimension, x1 and x2. But now, if you're large in X1, you also tend to be large and positive in X2. And if you're small in X1 or negative in X1, you tend to be negative in X2. So now there's covariance as well. So you can capture these correlation parameters and these variance parameters together in the so-called covariance matrix. And when you generate a multivariate normal, then you have to specify both of these things. If the covariances are all zeros, then this is just a convenient way of drawing from multiple independent normal distributions. But if your covariances up here and down here are non-zero, then you create dependency among the dimensions. So that's what I mean by a covariance matrix, just a little review. So the idea here is that we sample from the normal. We have the mean is going to be updated from the top R individuals from the previous generation. So we'll see how that's done. But for now, we just view it as like an average of those top R individuals. And then the covariance and step size is estimated from the previous steps. And so this is information from the previous generation, but it's also information about what directions have been successful in the past several generations. So behind the scenes, CMA-ES is keeping track of historical data, and it's using that to enhance the shape of this covariance matrix to help us guarantee that we're going to be sampling in productive directions. So what does that look like? Okay, so we sample those points, and then we're going to look at the shape and location of the top R points, the top R samples in this population. And I'll show a little example of this in just a second. And then we're going to take a weighted combination of those top R individuals that are weighted by rank. So if you are the highest ranking individual, you will the new normal, the new mean of the new normal will be more like you than the others. So it's a weighted additive combination of those top R individuals. So it's like a weighted recombination. It's going to amplify the influence of the best solution in those top R individuals of those samples. And then for the covariance matrix, we are going to look at the shape of those top R individuals, and we are going to grow members of this matrix, of this covariance matrix, toward the kind of extended areas. So the areas where those points are more spread out, they're going to pull step sizes to be larger. And the areas of those top R that are more compressed, they're going to tend to compress those step sizes. Now, we are also, and this is a key thing that is very different from NES, going to look at the accumulated movement directions over over basically the history of this algorithm. And we're going to use that to augment those variances in the covariance matrix to sort of give us some aspect of momentum. And that momentum is going to help us move in productive directions. So the idea here is that this normal distribution over time, as you update these parameters, will approximate the objective's shape. And so you can think of that over time, it's hopefully going to find the top of a fitness peak, the top of a fitness peak, in these multiple directions. And it's going to start approximating the curvature where this is the mean and these are the covariance matrix at the top of that peak. So so by sampling from the approximated shape of the top of that peak, what we're doing is drawing points that are more and more likely to be higher and higher fitness. That's the idea here, is that this over time is going to be a probability distribution of where to find the highest fitness. So roughly speaking, you could imagine that I'm going to draw M samples and focus on the top R. So at some iteration here, I might draw a certain number of samples like this. So they're all over this space. And so this space, I've got these two decision variables, X1 and X2. And then I might find that after I sample all those, that the ones that have the highest fitness happen to be the ones that are, let's say, I'm just going to sort of pick these kind of at random to try to make a point, are these. So it just happens to be that these points are a higher fitness than these. So you can kind of imagine we're sitting on a mountain that has kind of a ridge, and this is downhill from that. So these are all lower on the mountain in terms of fitness. And this is sitting up here on a ridge of this fitness plateau. So what this is telling me is that it would be useful to explore along this dimension, and it's not as useful to explore as far along this dimension. So we can walk in large steps along the ridge, but small steps kind of perpendicular to the ridge. Now, we happen to also know, and so this is what our the information gradient is. It will tell us not only where the highest variance is, but it'll also generally tell us that, let's say that the natural gradient or the CMA-ES approximation to the natural gradient is roughly in this direction. So this tells me that when I choose my step sizes, then I should choose step sizes that are reshaped so that this distribution is going to shift its mean, and it's going to shift upwards in this direction, and it's going to compress here so that in the next generation I'm more likely to draw points over here. And those points may be spread out in this direction, but they're going to be narrow, very tight in this direction. And so that will then gradually, then hopefully I get a cigar of points the next time through that are like up here. And then from that cigar of points, then we do this process over and over again. And you get, and then hopefully this moves and moves and moves until you find the top of this peak. So it's kind of like I found the ridge, but this ridge is continuing going up. But eventually I'm going to find this top peak over here. And then it should collapse around that point so that the whole thing collapses to basically a point. So that gets us to what happens eventually here. This CMA will anisotropically collapse to a fitness peak. So what that means is the distribution that it's fitting, it's going to look like a funny shape. It's going to look like a cigar where it's going to have some directions which allow for larger step sizes than others. But the whole thing is going to get smaller and smaller and smaller once you reach the top of that peak, because it's going to, in order to reach the natural gradient, to reach towards higher and higher levels of fitness is going to cause the step sizes to scale smaller and smaller and smaller so that the whole thing collapses down to like a single point. So the region of most informative upward steps shrinks to a point. And this is a lot like good old-fashioned gradient ascent with adaptive step sizes, but this isn't being set by gradients, it's being set by statistical properties. So by monitoring the shape of the covariance matrix and the fitness progress, what's cool about this is eventually we can say, this distribution is way too narrow. I can stop my sampling, and I can either just stop the algorithm there and say, I found the top of the fitness peak, or I can trigger a restart, a reset of the algorithm, where I just remember where we are, save that away as a good candidate, and then reset the population to a random spot elsewhere in the fitness landscape, and then try this process over and over again. And then and CMA-ES is particularly good at this because it tracks historical evidence. So it can really keep track of like, wow, we haven't made any progress for a very long time. We must be at the very top of this peak. So it's a good time to trigger that reset. So we can think of ES populations, particularly CMA-ES, as a type of depth first search in continuous space when you've got these adaptive restarts. GA populations are a type of breadth first search where we maintain multiple fitness peaks simultaneously, but we don't necessarily exploit them one at a time. We kind of try to exploit them all simultaneously. So we spread out, we get as much coverage as possible. The ES drills down or drills up to try to find a fitness peak. And then with adaptive restarts, you can try to find more fitness peaks, but it's really well designed to exploit one fitness peak at a time. So remember, these are kind of analogies because these aren't discrete, these are continuous spaces, but that's what you kind of think of as DFS and BFS. All right, so takeaway messages. Don't want to keep us longer here, so I'm just wrapping up. And so the idea here is that GAs are global optimizers. And so we have, they're basically hypothesis testers. So we sample a bunch of the population. Those are hypothetically good solutions. But then based on their fitness, we retain some, we discard others, the same way we do with evidence for hypotheses based on their relative performance. So plurality is possible, just like it is in hypothesis testing. Multiple peaks can be explored simultaneously, just like two hypotheses can be failed to reject simultaneously, and they might both be true at the same time. They might both be good hypotheses. Contrast that with ES. ES is not a hypothesis tester. It's a local searcher, which could be kind of turned into a slow global searcher with the adaptive restarts, which is much more like response surface methods, another framework in statistical or in design of experiments and statistics. So the population drives parametric changes to a model of the objective, and that model allows for more efficient, tailored search patterns where you can make mutations that have large step sizes in some directions and small step sizes in others. This is very similar to gradient methods, but they're based on statistical performance, not on the actual gradient information. So the model parameters allow for you to do these intelligent restarts, which give you that kind of breadth first search, depth first search, breadth first search, depth first search interpretation. And I should emphasize that GAs can be used generically on any decision space, whereas the CMA-ES are really stuck with these real value decision variables. Now, once you've come to a local spot that looks pretty good, then you can hand it off to good old-fashioned stochastic gradient descent or ascent, which does the kind of local fine-tuning. So if you have gradient available, you can use that gradient to fine-tune within those discovered regions. But you can't use gradient ascent or descent without a gradient, for one, and there's a bunch of problems you won't have a gradient, or with, and this kind of goes along with without a gradient, with generic decision variables like integer decision variables or things that greatly change the shape of the fitness objective. So for example, you can use stochastic gradient descent with artificial neural networks, but you really need a regularizer added into that loss function to sort of tame the gradient descent into one small location, and then a fixed topology. You kind of have to decide ahead of time. I'm using this type of neural network, and I'm not gonna change how many weights there are. But once I have all those weights, I'm going to tune those weights. These methods actually allow you to choose which weights to use and which weights not to use. They change the topology of the network. So you can think of like broad search over topologies of a network. You can think of then local search to sort of find to more fine tune within the plausible topologies found at the GA. And then once you've found a very good topology, then you can fine tune within that topology. So, and this is actually used. You can combine all three of these. This is very often done manually, where if you're in a problem like this, you might start with this one to get you in the ballpark, explore those regions that you've discovered, and then from there, fine tune a bunch of solutions that you feel are pretty good. And there are sort of automatic ways to do this too. So there are machine learning approaches that you can kind of look across all of the different ways you could solve a particular modeling problem, and they will use these methods to come up with a type of model to use, and then the parameters of those models will gradually get fine-tuned through these methods. So they're complementary. They aren't replacements for each other. So there are things that GAs can do that gradient descent can't do, and vice versa. And the best thing that you do for a generic complicated problem is combine them. All right, so next time, like I said, I want to stop here because we're at an hour 15. We just talked about a very different way to sample populations to make progress in engineering design problems. We are going to now diversify that and look at, rather than a traditional optimization problem, how do we turn programming itself into a type of optimization problem that methods like genetic algorithms can end up solving. So how do you tell a genetic algorithm, write me the best piece of code to do X or Y, and then have it write the code for you. And then after that, then we'll go into immunocomputing, where we get rid of a single objective and we instead shift to solving the problem of maintaining a diversity of possible solutions. And these will end up being possible classifiers to effectively unknown problems or unknown threats. And so this will be inspired by the adaptive immune system, which itself uses evolutionary approaches, but they're not from generational perspectives. They're inside your body at a life history perspective. So they're a bit more real time. So that's where we're going. So overall, this unit is about different ways to use evolution to inspire variable samplers in these decision problems and in machine learning applications. All right, that's long enough. So we will see you back in person on Thursday. And if you have any questions, please feel free to reach out.